Well, thank you very much for your kind welcome and for permitting me here to talk to you tonight. First of all, Dr. B, honorable consuls and chairpersons of the various associations between Heidelberg and the United States and Germany and the United States, honorable guests, ladies and gentlemen, when I talk today about something which we feel provides some new information on causes of some very common human cancers, I do it jointly with my wife, Esther Michel. You mentioned it a moment ago. She's always saying that she does the work and I do the talking, which is not exactly true because she contributed a large number of good ideas and quite a number of nice hypotheses as well to the story. So she's really a full-term scientist up to today, I should say. Now, I'm going to talk today about an interest of mine which is existing since for my whole lifetime. As a medical, studying medicine and becoming a licensed physician even at the end, I had decided very early, prior to studying even medicine, to go into research later on, and that I did. And for my whole lifetime, I worked on infectious agents linking to cancer. Why? Because during my studies, I learned about bacteriophages, about bacteria picking up some genomes of bacterial viruses. Those genomes persist in those cells, modify the properties of the respective cells, and even in some cases cause a pathogenicity of the same agents. And that story fascinated me from the very beginning when I heard about it. And the idea was already at an early stage, is that something which has a relationship to cancer? And today I will tell you a story which really relates to this original conception of infectious agents and cancer. As far as my experiences in the United States are concerned, I spent only 3 years in Philadelphia, but these were 3 very fruitful years and a long period ago in the 1960s, from 65 to 69. But it was for me quite an important period in my scientific life as well. Now, let me just start out by mentioning a few aspects of our work. You will see later on why I have this specific picture here on the first slide. I will not mention it right now, but it is important, as you will see later on. The present state of cancer globally is that we have basically everywhere an increase in incidence of most of the cancers, not of all of them, but clearly of the majority of cancers everywhere in the world. And at the same time, we have a decrease in mortality, a slight decrease in mortality, which doesn't match really the increase in total. And it's an interesting question. Although, indeed, a lot of progress has been made in treating human cancers, why can we not cope with the increase of these types of cancers? And my favorite theme is really, why don't we do more in prevention of the occurrence of cancers? Why don't we start early and prevent some of these diseases, like the anti-smoking campaign and many other things? I will not go into the details, but this is an important issue. And if you look into the present expenses of the governments for prevention in comparison to treatment of patients, and I'm not saying anything against the treatment, of course, you will see that there's an enormous discrepancy in funding. It's for prevention, usually in best countries, it's about 4% of the expenses for health-related reasons. Now, since a couple of years, or since quite some time, we know indeed that approximately 20% of the global cancer incidence is linked to infectious events. The mode of action is quite different between these infectious events, but the identification of such agents which are linked to human cancer, specifically hepatitis B virus to liver cancer, the papilloma viruses to cancer of the cervix and a few other cancers, Also of the bacterium Helicobacter pylori to gastric cancer, the parasitic infections in specific countries, a major problem, and also the HIV epidemic leading by immunosuppression to some increases, tremendous increases in certain types of cancer, that helped really to develop new means of prevention. Prevention, of course, optimally by vaccination to prevent even the early symptoms of those infections. And secondly, also by treatment, like the bacterial infections, the parasitic infections, they can be effectively treated. And more recently, hepatitis C virus infections, a major cause for liver cancer outside of the endemic regions of East Asia, can be also treated quite effectively by chemotherapy, indeed. So in a way, the identification, and particularly AIDS, I forgot to mention, of course, the therapy of AIDS does not cure the infection, but it reduces the symptoms and it reduces the load of the infection and the side effects of the infection. So the identification is important for prevention, quite important. And that is something which we try to analyze throughout my lifetime. We can basically know now a few modes by which infection act in leading to cancer, and I've listed them here. And basically, we discriminate between what we call the direct carcinogenesis. Direct means here that genetic material of the respective agent persists in the respective cancer cells, or even the pre-cancer cells leading to cancer eventually. And usually some of these genetic information is expressed in those cells continuously. In cervical cancer, if you block the expression of the papillomavirus genomes which persists there, then you can render the cells virtually non-carcinogenic. They're not normal either, because they acquired already some modifications, but they're no longer true cancer cells. Well, besides these, there are a couple of other aspects which for time reasons I will not dwell on right now. There's one other mode of interaction which became only clear relatively recently. Namely, of course, I shouldn't say the virus-induced immunosuppression did not become clear recently. It's known already since quite some time that it activates other latent infection, tumor virus infections as a rule, which increase under immunosuppression quite substantially, like the so-called Kaposi sarcomas in Africa, and a tremendous problem due to AIDS infection. Or also the so-called Merkel cell polyomavirus. Merkel has nothing to do with our present chancellor in Germany, but it was a pathologist in the 1990s, 1890s, who discovered a disease which in Pittsburgh by Moore and Chang in 2008 has been discovered to be the reason for this type of infection. So immunosuppression plays a role, but the other one, and that is the one I will dwell today exclusively on, that is agents which induce at specific spots chronic inflammatory lesions, and those specific spots lead to the production of chronic inflammation, of the production of some radicals. Radicals are in some way modifications, inducing modifications in the genetic material, mutations in genetic material, and modifying the cells which may not necessarily carry the virus or the agent. For hepatitis C, it's quite clear already since quite some time that in the liver, you don't find the hepatitis C virus in the cancer, which develops after 30, 40 years later after the initial infection, but you see the viral genomes only in the inflammatory lesions around the liver. So that is a theme which I will discuss today to some degree, and the other things which are listed here on the slide I will not mention right now because they do not seem to play a significant role in human cancers right now. There's one striking aspect which was clear from the beginning, namely that many of these infections take decades. The onset of infection at early age frequently, development of cancer in some instances decades later. There's a few exceptions from this rule. There's the one which is the upper one here, the Epstein-Barr virus-associated X chromosome-linked lymphoproliferative syndrome, a complicated name, but there you have already an inherited mutation in your genome, and which was fostering, of course, the view that mutations are essential for the development of cancer. And to some degree, this is still maintained up to today, with some justification, of course, as well. There are other modifications, other infections, which have shorter latency period, as we call it, between infection and cancer development. For instance, in Africa, relatively frequent Burkitt's lymphomas, specific types of Hodgkin lymphomas, and the Kaposi sarcomas, due to HIV infections, they require shorter latency periods. What we know today is that none of these infections which are listed here on this table is by itself sufficient for cancer induction. You need indeed always some additional modifications in the host cell genome, in specific spots of the host cell genome prior to the development of cancer. And the latency period itself is determined by the number of additional mutations which have to be acquired prior to the development of cancer. With other words, what you see here in hepatitis B-linked hepatocellular carcinomas and C-linked carcinomas, parasitic and bacterial infections, they require quite a number of modifications in addition to the infection prior to the development of cancer. So the latency period reflects this kind of interaction between an infection on the one hand and genetic modifications of the host cell on the other hand. Some viruses are able to induce the mutations themselves. In other instances, tobacco smoking, of course, is all of you well known. Also, the viruses which might be involved as well are not well known, is an important example here in this case. Now, so since the development of the Human Genome Project, a large number of cancers, human cancers, have been totally sequenced. The nucleotide sequences of individual cancers are known. For many of our common human cancers, individual cancers have been analyzed. And yet, since the year 2008, when Mu and Chang identified the Merkel cell polyomavirus, no other cancer popped up, in spite of a steadily increasing number of totally sequenced cancers, which seem to directly linked to an infectious agent. This has, of course, two consequences. Either we know all these agents, which are linked to human cancer, quite a possibility, or the alternative is that the way how these agents cause cancer is somewhat different from the conventional way of, let's say, I call them frequently conventional tumor viruses, like papillomavirus, polyomavirus, and many others, Epstein-Barr virus, and so on. And so we decided, when I say we again, my wife and I, as Michel Devilliers, we decided to look into the second question. Is there a possibility that something else is playing a role which is different from the conventional way? And I will briefly give you a little bit of an historical background of this whole story, because it's known since a very long time, namely exactly since the year 1978, when Sugimura, in particular in Tokyo, analyzed specific types of meat, that colon cancer is linked to some degree to what they called at that time the red meat consumption, because first of all there's a higher incidence wherever people consume for longer periods of time red meat. Red meat is beef, sheep, and pork, and a couple of other meats, and also processed meat, smoked, air-dried. A couple of other processing modes exist still. That seems to be linked to a higher cancer risk. And countries with the highest rate of so-called red meat consumption, in particular Argentina and a couple of others which are listed here, including the United States and most European countries, they commonly reveal a high rate of colon cancer. That story intrigued us from the beginning, because first of all, when Sugimura in 1978 reported that in the roasting, grilling, barbecuing, and frying process of meat, specific chemical compounds are being produced, which he claimed, and it's proven, that they indeed have a carcinogenic function. if you provide these kind of chemical carcinogens to mice, for instance, usually at doses which are about 10,000-fold higher than the doses which are presented in the red meat itself, the grilled red meat itself, they produce tumors indeed. And so for the majority of, particularly of epidemiologists, and that is true up to today, these represent the basic factors linked to the etiology of colon cancer. And in a way, as I wrote it here, it sounded so reasonable that it blocked alternative explanations for quite a while, and even attempts to publish those alternative explanations for almost 30 years by now. Interesting point, and I wrote here the sentence, dogmas and prejudices obstruct the vision, occasionally the vision of the realities, and I'll tell you why a little bit later. Sorry. Now, what was striking us was from the beginning the geographic epidemiology of these types of cancers. That was a cancer, in particular cancer of the breast and cancer of the colon. All these dark-colored areas here, these are maps which are provided by the International Agency for Research on Cancer in Lyon, the World Health Organization. The dark areas reflect the areas with a high incidence in the upper part of breast cancer incidence, in the lower part of the picture of colon cancer incidence. And there's not in 100%, but a remarkable coincidence of frequency of both types of cancer globally. So there must be some kind of a specific reason in those countries that they have this higher frequency of cancers. There are other cancers where the incidence is extremely low. And the most striking example is Mongolia. Why Mongolia? Because Mongolia is one of the countries with the highest rate of consumption of red meat, grilled, barbecued, and in large quantities. And that didn't fit into the picture at all. India is, as I always say, a paradise for epidemiologists because their specific types of meat are not being consumed. Which types? Everyone knows it. Beef is not in India permitted to consume. Bolivia is one country where they eat meat, and they eat a lot of meat, but they eat a very specific type of meat, also of beef, different from our, at least from our Eurasian type of cattle. To make a long story short, in fact, for us, this suggests that it's not red meat per se, and that you could study quite nicely from a few other places in the world, from China and Japan and Korea as well. But it must be a specific type of meat which bears a risk for developing these types of cancer, at least at high frequency. And we suspected that this is our typical dairy, Eurasian type of dairy cattle, aurochs-derived dairy cattle, which is so popular in Europe, everywhere in Northern Asia, and also in North America as well, and in Argentina. So we started out with a speculation. This whole risk of red meat is something species-specific for certain types of cattle, the dairy cattle, the Eurasian type of dairy cattle. If that is so, I'm a virologist for all my life, and I know that species specificity is frequently a sign for an infection. There are a couple of infections which are highly species-specific. So that started the speculation, do we deal here with a kind of infection carried specifically by a specific type of meat, namely here of our dairy cattle? Wherever in countries different types of cattle are being kept, for instance in Mongolia with the yaks, very different type of cattle, genetically different type of cattle. They also have an endogenous type of cattle which was genetically different from our dairy cattle, but crossbred it after the Second World War in particular with zebu cattle. In other countries where zebus are being maintained, the rate of these cancers seem to be at least relatively low. Wherever our Eurasian type of cattle is being kept for milk production, meat production, not so much, but milk production in particular, the rate seemed to be elevated and in some instances even extremely high. There were striking observations which have been made, particularly in Japan and Korea, after the Second World War and after the Korean War. It's on the right-hand part of this picture. After the Second World War, enormous amounts of beef, but also of porks, have been introduced from the United States to Japan. And even raw beef is popular to some degree in Japan right now for quite a number of meals. In Korea, it started about 15, 20, 25 years after the Korean War as well. And the same had happened there. There was an enormous increase, as you can see in the lower part of this picture, in meat consumption in Japan and Korea from the year 1961 to the year 2002. And concomitantly, there was a remarkable increase, in particular in colon cancer. There was also an increase in breast cancer, but not as steep as elsewhere. For India, the data remained relatively constant, a slight increase, but not very significant in that case. There was another thing which didn't fit into the whole story of chemical carcinogens. Namely, if you grill, barbecue, fry, or whatever you do with chicken or with fish, you get the same chemical carcinogens produced as in a well-barbecued steak or air-dried steak and so on. Yet, the data which existed from epidemiological studies indicated that chicken is considered as quote-unquote safe. There's no increase in colon cancer incidence in those cases. Fish was even considered to be healthy, protective against these types of cancers. Long-time fish consumption, at least, seemed to have an effect. This, again, didn't fit into the whole story. So there were a couple of inconsistencies which helped our kind of considerations, but were quite ignored and neglected by many of the epidemiological studies. So for those reasons, and for one reason, because at the end of the 19th, and having worked for 30 years on papillomaviruses and human cancer, both my wife and I, we became both very tired of this type of studies. There was not very much new coming out of it. The industrial companies took it over and made a lot of propaganda with their vaccines, of course, and we decided to change the topic completely. And we did it, and she did it in particular, working for quite a while, for almost 10 years, on so-called TT viruses. TT viruses were discovered only shortly before that period of time. These are small, circular, single-stranded genomes which are found almost in every human adult. So we are all infected by those agents for a lifetime, most likely. And so we suspected at that time, since we knew something like this from Epstein-Barr studies, that the situation was quite similar here in that we have a common agent which causes a few very specific types of cancers. And so we started with techniques which were developed in the TT virus studies where we didn't find a connection to human cancers, where there's something in cows. Serum of cows, 120 sera from cattle, dairy cattle, we received from the University of Leipzig, from a veterinarian station there, and about, in addition to milk preparations from the supermarkets around Heidelberg, yogurt, milk, and a couple of others, milk products, cheese, and so on. Well, the results were amazing from the beginning. Looking specifically now for these types of single-stranded circular DNA-containing agents, we found quite a large number of them from the beginning. These were, what's shown here, were the first data which we analyzed. We differentiated them into two groups. We called them bovine meat and milk factors, BMMF, 1 and 2. The group 1 was characterized while they have a large open reading frame for a gene coding for a larger protein, which is quite similar between the different types which we've isolated, even those which came later in addition. And the group 2 is usually larger. They contain usually 2 large open reading frames. And so they became interesting from the beginning. The disturbing aspect was initially that virtually all of these sequences had some relationship to bacterial plasmids. So bacteria, if you see something bacterially related up to today in total sequencing of human tumors, it's instantly suspected to be a contamination, and they are thrown out and not further considered. Well, due to the previous work on TT viruses, we didn't consider this, although we were disturbed by the similarity. And we saw that these sequences, when we brought them into human cells, they became genetically active without the bacterium by themselves. And in specific lines, they also started to replicate under those conditions, independently, autonomously, in those types of systems. And that triggered our interest quite a lot. In addition, we got 2 of those sequences from the analysis of an autopsy lesion of a patient with multiple sclerosis. It was 2 different types from the same patient and the same lesion, but that triggered our interest even more. This shows here the analysis of our genetic activity, the early analysis which we did. Some of the co-workers who did mainly the work are listed there. We saw clearly the transcription of DNA, and we also could demonstrate with antibodies which we developed later on in the game that the transcripts are also translated into proteins, which can be seen in such specific cells. Well, we followed up the story of multiple sclerosis for quite a while, particularly with the help of the Swedish group of Thomas Olsen and Hans Grönland. And we could demonstrate that against this group 1, with a large open reading frame, there was a significant increase in antibody titers against the agents of our so-called BMMF1 group, of the bovine meat and milk factors 1 group. And that triggered our interest even more, because for that type of disease, there's a long-standing connection with Epstein-Barr virus infection. And we had done in the past some experiments which demonstrated that Epstein-Barr virus, a large viral genome, if it infects the cell, similar to other herpes group viruses, and if the same cell is infected by a couple of other agents, single or double-stranded small DNA viruses, for instance, there's an enormous amplification of these small other agents under those circumstances when the herpes virus becomes reactivated under those circumstances. So it triggered an idea that we have this type of very interesting synergism between herpes virus infection, specifically an Epstein-Barr virus infection, and one of our BMMF isolates. I will not discuss this to any more detail today, because it's an ongoing project at our place still up to today, but it's very interesting because it has been shown that Epstein-Barr viruses are particularly induced under vitamin D deficiency. And under those circumstances, which was studied in particular by workers in the Antarctica who were not exposed to sunlight under those conditions, they start to produce large quantities of Epstein-Barr virus in the saliva because they're all latently infected, and Epstein-Barr virus becomes reactivated in those instances. And we speculated that this is one of the reasons why in infectious mononucleosis, the synergism leads to the production of some unknown agents. In this case, we suspected our BMMF agents, and that they are the ones which now either as proteins or particles infect the surrounding cells in the brain and lead to the lesions. One of these lesions is shown in the early lesions in the lower part. I got it from the neuropathology department of Los Angeles University, and that the pathogenesis is based on the interaction of 2 different types of viruses under those circumstances. As I said before, in multiple sclerosis, we do find significantly elevated antibodies against the major protein here of this agent. And in addition, one finds in multiple sclerosis indications for Epstein-Barr virus reactivation, and the specific antigen of Epstein-Barr virus is consistently elevated in those cases in the sera of these patients. Well, as I said before, I will not go into this in more detail. It was for us an interesting story, which has some interesting follow-up as well, even for other neurological diseases. But let's come back to something else which interests us, particularly for colon and breast cancer. Namely, there were some long-standing observations that prolonged breastfeeding is protecting against a number of known infections, which do not occur as long as these babies are being breastfed. And these are, for instance, diarrhea infections, respiratory tract infections, which in India play a major role for the mortality of newborn babies if they cannot be sufficiently breastfed under those conditions. Rotavirus, norovirus, the most frequent causes of these types of diarrhetic type of infections. The human polyomavirus type 9, yeast type of infection, Candida albicans, specific types of parvoviruses, and specific strains of influenza viruses. There's all protection going on as long as these babies are being breastfed. There are a few other conditions which evoke some interest, type 1 and type 2 diabetes. A couple of publications describing this, and also inflammatory bowel disease. Well, that was interesting because in human milk, there are specific types of sugars, which are listed here, the so-called 2' and 3' fucosylactose, the left part, the upper two, and also a rather complicated molecule, the tetraose, which seem to be more or less selectively present in human milk. There are a couple of other of these types of sugars also in human milk, but these seems to be clearly the three predominant ones, and they are so predominant and so considered as protective that they're even given right now into formulas for babies which cannot be breastfed in order to provide some kind of protection as well. What they do apparently is they block on the surface of some cell membranes some conformations which act as receptor stations for the uptake of infections in this case. That is relatively reasonably well established. You can see it on the lower part of the slide. The 2' and the 3' fucosylactoses are differently expressed in the postpartum interval, and in fact the one goes down after prolonged breastfeeding, the other one goes up during the same period. So this was quite interesting because not only the infections, there's a protective effect against infections, but there's also frequently a protective effect reported against some malignant diseases. Specifically for Hodgkin's disease, it's not so easy to follow it up because it usually occurs in age groups much later, between 15, 25 years or so of age. Non-Hodgkin's lymphomas, a couple of reports describe this. Acute lymphatic leukemias for babies, there's a protective effect, relatively frequently reported, and also less frequently reported for neuroblastomas, nasty disease for small babies. And also there are reports claiming protection against multiple sclerosis by this type of prolonged breastfeeding. And this is now an interesting observation which I particularly studied here in San Diego by Dr. Baki, who I'm happy to see him here sitting in front of me, and his crew, on specific leucans which are present in human milk and not present in cow milk. There's one specific N-acetyluramic acid, the so-called NOI5AC, which is present in human materials as well as in animal materials. But in humans, we have a defect in one enzyme, which is listed there. I will not pronounce his long name right now, which is rendering this enzyme defective. And humans cannot convert now this NU5AC into NU5GC, an N-glycolyluramic acid, which you see on the right-hand side of the slide. So this type of modification seems to be a very important one in the whole story, because humans are unable to synthesize the NU5GC, but animals do it. And as shown here by the San Diego group, but also by other groups by now, it's quite obvious that the nutritional uptake of NU5GC from cow and cow milk products leads to an uptake of this molecule into humans and to an incorporation almost exactly in those sites which previously were blocked by the sugars for the uptake of a couple of infectious agents. And under those circumstances, if now this animal U5GC is taken up, it renders them susceptible for a couple of those agents against which the babies were not susceptible at all. And in addition, of course, it raises an interesting question, what other effects this incorporation may have. So the dietary uptake of Nu5GC after weaning from dairy products and its incorporation into human membrane receptors changes the properties of the latter, and they become now susceptible to those infections previously during the breastfeeding period blocked by the specific sugars, and apparently also by a couple of other ones as well. Well, the well-established effect, or protective effect, of breastfeeding for newborn babies raises instantly an interesting question. Do the sugars which cause this effect in neonatals also protect the breastfeeding mothers? Because they're produced in the breast and they're excreted in the urine of the mothers for a certain period of time, as long as they are being produced, so they must somehow auch pass the bloodstream, leading to the excretion in the kidneys. In particular, do multiple breastfeeding periods affect the incidence of certain types of diseases? Obviously, with the speculation that this may be linked to some kind of infections. Specifically, of course, the question, are the cancers reduced under those conditions? And could this be due to an infection-preventing effect? The answer is surprisingly well studied in the literature. It's available everywhere since years and decades almost. Namely, the answer is yes. There are a couple of cancers which are protected after multiple deliveries. This is a large study which was done to part here in the United States, which showed in, if I recall it correctly, 122,000 multiple deliveries here, that indeed those mothers who had multiple deliveries here in this graph here showed a 36% reduction of breast cancer incidence following them up after a long period of time. Other studies, and there are a couple of other studies also available, come up to 50% reduction of breast cancer, but usually the numbers were smaller which were studied. Colon cancer goes down, lung cancer goes down, interestingly, and even ovarian cancer goes down. And so there is an effect which was striking for us. This is schematically outlined here. The shaded periods in the bars there should indicate the periods when these types of sugars are being excreted, the fucosyl lactoses and the tetraoses during the various pregnancies. And during this period, they're of course present in the respective mothers as well. And they're excreted, as I said before, in the urine under those circumstances. There are other interesting data along those lines, also known since quite some time, not new really. Namely, this is a study here in 153 countries between 1995 and 2013 on the distribution of breastfeeding for at least 12 months or less in these instances. The data are quite interesting if you analyze them carefully. The dark shaded areas here indicate all the areas with prolonged breastfeeding, when average 12 months of breastfeeding are customary. You can see that countries like Mongolia, India, Equatorial Africa, Bolivia, they show a lot of prolonged breastfeeding periods under those circumstances. And as I showed you before, and I think I have it still in the next slide once, there's the lowest rate of the respective forms of cancer, of breast and colon cancer in these cases. There are few countries which are just on the opposite side, specifically Britain and Canada, to some degree also Holland, in addition, maybe to some degree Sweden, but the data were not so clear there. And those countries are the ones which, at least within this hemisphere, have the highest rate of breast cancer and colon cancer. Such statistics do not prove anything, but they look interesting in regard to what we speculated on. So let me summarize this here. The importance of dairy factors as risk factors for specific cancers is underlined by the countries with the highest rate of breastfeeding 12 months after delivery in average. They commonly reveal low rates of colon and breast cancers, and conversely, countries with the lowest rate of breastfeeding usually exhibit high rates of colon and breast cancer. In the Swedish study, which was conducted from the Swedish Cancer Registry, on 23,000 persons who are lactose intolerant, which means they do not consume dairy products, at least avoid them as much as they can, and comparing them to 70,000 siblings in the same families which are lactose tolerant and could tolerate very well lactose. And the parents, either parents or siblings, revealed reductions in lung cancer. Again, lung cancer, where everyone knows that smoking plays a significant role, by 45%, ovarian cancer by 39%, breast cancer by 21%, among the lactose intolerant individuals. So lactose intolerant has some kind of protective effect in this respect. So, well, for all these reasons, we started relatively soon. I hired a young postdoc 4 years ago and asked him specifically to develop monoclonal antibodies against one of our common proteins, the large common protein in our BMF1 group. He did it very skillfully, and we came up with a collection of, by now, 5 distinct monoclonal antibodies which cover distinct antigenic epitopes of the same protein, of the same large protein. And this is schematically outlined here. The bar outlines the full length of the protein, of the coding sequence of the protein. And you can see that in the first part of the genome, the N-terminal, as we call it, part of the genome, there's a rather very high rate of consensus between the different isolates which we obtained of about 86, 87%. The middle part is a little bit less conserved. It's about, what is it here? Something like 76%. And the right-hand part, the so-called C-terminal part of the protein, there's a conservation only of the genome, there's a conservation only of 37%. So it differs very much among different isolates. That has some practical importance in the analysis of the persistence of such genomes in tumor cells. Now, by using these monoclonal antibodies, we came up with a surprising observation, which we initially thought is something funny and maybe even a kind of specific contamination. Namely, what we saw here was really that the colon, the crypts here, the so-called Lieberkühn's crypts, as they are called, let me see, I have to handle it the wrong way, which I'm afraid you will not see it perfectly. These are the cells from which colon cancer arises. And you can see here that they have nuclei. That's important for those who can sit close enough to see it. The nuclei all are located at the periphery of these types of cells, typically. They are not stained with our monoclonal antibodies, but the surrounding tissue, the so-called lamina propria, is intensively stained here. The foci of such cells, which are heavily stained by the monoclonal antibodies against our, which is directed against our antigens. There are a couple of pathologists who helped us from Zurich, Achim Weber, from Heidelberg, who's an epidemiologist basically, Hermann Brenner, but also the immunologist Matthias Heikenwälder, who helped us with the development of these techniques. Anyway, it was surprising. Why don't we find it here in the cells from which cancer develops? We looked into cell lines which originated from colon cancer and from breast cancer. They were all negative for what we were searching for the nucleic acids. Of course, there was one other feature. We started then, what else is in these types of lesions? And it became quite clear that the same parts which show this foci, which is shown here, these labeled parts here in the cells, are also stained for inflammatory signals, namely for the so-called CD68 antigen demonstration, which is clearly an indication of macrophage-mediated inflammation there. So there's an inflammation going on in these regions that's now repeated on and on and on. It is consistent everywhere. That the same lesions are stained by these totally different types of immunological controls. And the lower part of this picture shows what is interesting here. If you see these crypts, which I showed you before, in a cross section, if you see it here in this type of tangential section, here's the lumen of the colon. The crypts are opening to the lumen, fulfill their secretory functions there. But in the crypts themselves, there are enormous numbers of cells in the lower part of the crypts up to the middle part of the crypts who are consistently replicating DNA. They are replicating actively. They migrate to the upper part, differentiate out, and fulfill their functions there. So there's a continuous rate of replication. This is a kind of cross-section here. Wherever you come closer to the bottom, you see more of these cells. In the upper parts, there's no replication anymore visible under those circumstances. So this raises the suspicion, isn't there something which we suspected already for a long time for hepatitis C virus infection, that there's an inflammation going on not in the material from which colon cancer starts, but just in the adjacent material, which produces something like oxygen radicals, mutagenic products of inflammation, which may act specifically and intensively on cells which replicate actively, because those mutations are being fixed under those circumstances, which are not fixed in parts of the cells which do not replicate under the same circumstances. Whereas these foci are themselves, you find very rarely, at least 100-fold less replicating cells in those regions than in comparison to foci. So this would be for us a classical example of an indirect carcinogenesis. Then we started to look whether these oxygen radicals are really formed. And there are tests available for this. And this is a test which is done with this 8-hydroxyguanosine, which is a modification which occurs under the function of oxygen radicals. It's commonly used for the demonstration of oxygen radicals. And the yellow staining here, I don't think it comes out very, very well here, maybe on this side better. The oxygen radicals are clearly formed under those conditions. Even with other methods, we can confirm it now. They are there. So we have indeed mutagenic events taking place in the close vicinity of the Lieberkühn's crypts. And as I pointed out before, where the nuclei, the replicating nuclei, are all located at the periphery of these crypts. So this should be really easily vulnerable for those types of mutational events. And that was the slide which I had in my very first slide initially. These are arbitrary stars which are included here by assuming that those are the places where the radicals are really active and modifying the cells. The consequence of these kinds of observations and considerations is that we have a specific trigger, not inside the tissue which develops into cancer, which induces random mutations at cells which proliferate very rapidly in the very close vicinity. That's a major message. We have analyzed about slightly more than 100 of those sections from cancer tissue and normal tissue in these cases. We find these crypts in about, these foci, in about 75 to 80% of all these which we investigated. There are few, it's the right one here for instance, where the normal tissue besides the crypts and also the tumor tissue, which are presented in the lower part here of this slide from each of these sections, which are both negative. We suspect at this moment, since our monoclonal antibodies do not cover all the agents which we've isolated right now, that they may contain similar, as we saw it in the papillomavirus story, that they may contain still other agents which need to be identified with specific monoclonals which we don't have available at this stage, but we are presently preparing. So basically, in papillomavirus studies, initially, we found in 50% HPV-16 and 20% HPV-18, which is more or less true throughout the world. But there are a couple of others which I later found, which brings the number to more than 95% at the moment. So the other interesting part of the story came up very recently. Again, the work which Esther Michel did quite effectively and very intensively, analyzing namely all the isolates which we have right now. This is here of both the so-called BMF1 and BMF2 groups, and studying their relationship to each other. What you see here, whenever the arrows or the individual scripts are very close together, then these agents are very closely related to each other, or if they are distant, they are only distantly related. You see that the BMF1 group on the right hand of this slide comprises a relatively homogeneous group. The BMF2 group is quite heterogeneous in this study. And the interesting part is another one, which I marked here in yellow, namely that there exists a specific type of plasmids in a specific type of bacterium, namely Acinetobacter baumannii, which shows a remarkable similarity to our isolates, with some of them very closely related to the isolates. And this type of plasmid has some structures which are different from the common structure of bacterial plasmids. They are different because, first of all, it has a so-called polyadenylation signal, which is quite unusual for bacterial sequences and bacterial messenger RNA. And in addition, there are also some structural differences visible. These are small single-stranded plasmids, similar to those which we have identified here, whereas the other ones, the other plasmids in the same bacterium are usually large plasmids, very different from this one. So we do not know where this group comes from. It looks to us, at least at the moment, that the bacteria may have picked up these types of plasmids. Some of these plasmids are, by the way, of importance for the antibiotic resistance transmission from one bacterium to the other, that it may have been picked up from some other organisms and somehow replicated very well in these types of bacteria, but they are the ones which were able to replicate in human, or being transcribed in human cells as well. So this is in itself a fascinating story of the evolution of those types of plasmids under those circumstances. It is an enormous type of work which was done here in identifying and sequencing each of them and comparing each of these sequences with each other. Here the middle part of this slide shows this one bacterial plasmid, the original type of bacterial plasmid, and the red dot there and the arrow indicates the polyadenylation signal in this plasmid which exists there, quite in contrast to the other plasmids, the other surrounding ones, and their number increased tremendously by now. So Michel has analyzed by now identified more than 20 types in the BMMF1 group, and more than 100 or slightly more than 100 types in the BMF2 group, which comprise the pattern which you saw in the previous slide. So it's a heterogeneous group of agents, and of course we have not analyzed too many of them right now for their potential pathogenicity in humans, but clearly they are widely spread and quite intensive. This is a new type, a new class of pathogens. They're not really viruses, and they're not really bacteria. They're plasmid-derived agents which learned somehow to infect and replicate in human cells. And if you would have sequenced those tumors initially and found these bacterial sequences, we probably would have thrown them out, as it is being done up to today in almost all sequencing assays for human tumors as contaminations or contaminants. But these are not contaminants. They are indeed present there, and they are apparently pathogenic. The other aspect, which is very interesting, we know also since a long time, published since a long time, that there exists a number of non-steroidal anti-inflammatory drugs, the well-known aspirin, for example, ibuprofen, and COX-2 inhibitors, which have a kind of preventive effect for a couple of cancers. This is a kind of a meta-analysis which was conducted some time ago, in 2005 already, showing the decline of the risk for certain types of cancer. For instance, if you daily take in 100 milligrams of aspirin or of some of the other drugs, a warning beforehand, don't do it without any type of control, because they lead to a higher tendency of bleeding and COX-2 inhibitors have been incriminated for heart attacks as a potential agents. So if that is being done, it should be done under supervision by some physicians. But nevertheless, the data which are available right now seem to indicate there's a 63% reduction for colon cancer, 39% for breast cancer, 36% for lung cancer, 39% for prostate cancer. And significant reductions were also observed for esophageal cancer, stomach cancer, and ovarian cancers. Now, what do these anti-inflammatory drugs do? They block inflammatory events, they block the production of oxygen radicals, and they block, obviously, the appearance of mutations under those conditions in the adjacent tissue. So this fits too beautifully almost into the whole story to be true, but we are by now firmly believing that these data are real. So what we say right now is there exist zoonotic infections, at the moment bovine types of infections, which we have characterized only, occurring during the first and possibly up to the second year of life. And they emerge as the main risk factors for specific cancers, here specifically colon cancer. The data for breast cancer are also positive, but we have not done as much studies on breast cancer as we did on colon cancer. And even for prostate cancer, there's some hints that it is a similar situation. All cancers with an unknown etiology so far. And they all show chronic inflammation, the induction of chronic inflammation that can be is known already since a long time to the pathologists for quite a while, but usually not too carefully further investigated. And they most commonly lead to these types of diseases after several decades following a slow chronic infection which is going on and on and on for years, inducing consistently probably low rates of mutations at any stage. But the mutations are measurable. And of course, colon and breast cancer contain plenty of mutations. Now, if you have an inherited gene already, which provides already a risk for breast cancer, the BRCA1 or 2 genes, for instance, or the AP1 gene for colon cancer, then of course you need less modifications in addition in order to develop the respective form of cancer. And those types of cancers in those persons develop at an earlier age and are quite devastating. So this, I'm coming to the end of my talk, this is basically our proposed scheme for colon cancer etiology, coming down from the cattle milk products and meat consumption, that the infection occurs after weaning during the first year of life, and that particularly the receptors, which are discussed before for reasons which we can discuss later on, the new 5GC receptors, containing receptors, play obviously a very significant role in the whole story here. That the primary target of these infections are, for some reasons, the mesenchymal cells of the colon. We cannot exclude that macrophages are also primary targets, but it seems at the moment rather that they invade the inflammatory reactions which develop there as a secondary event. That the invasion leads to chronic inflammation, a slow induction of oxygen and nitrogen radicals, inducing random mutations in adjacent proliferating cells of the Lübberkin's crypts. The precursor lesions develop. In the precursor lesions of colon cancer, we can beautifully demonstrate also the same positive foci, as we can demonstrate it in the normal tissue of colon besides the colon cancer lesions, and that they subsequently, in a period of 30 to 70 years, lead eventually, in average, to the development of this type of form of cancer. And the anti-inflammatory drugs, of course, would reduce the inflammation. They would act against the radical formation and therefore also reduce the rate of mutations. And that this is, so to speak, a nutritional infection with those agents after weaning and after systemic spreading to probably many other places as well. and that we have here is a specific trigger leading to these random mutations. We have inherited mutations, as I mentioned before, as additional risk factors leading to earlier period of cancer development. And we have probably occasionally a synergistic contribution of other mutagenic factors. Alcohol consumption and a couple of others may play a role here. But it seems so far that the evidence is relatively weak for those cases which we have investigated. But the latency period is here somewhere in the order of 30 to 70 years. Just to make a few conclusions, the three cancers which we are primarily investigating, breast cancer, colorectal cancer, and prostate cancer, account for about 30% of the human cancer incidence globally. In some countries to more than 30%, in others less than 30%, but in average about 30%. And if we add them to the list of known cancers which are linked to infectious events, we come up to slightly more than 50% already right now. So which shows that infections are indeed one of the major risk factors for cancer development. So here I'm making a few final statements. Epidemiologically, the story of red meat, of raw, roasted or cured meat, and of dairy products in human diseases has been, in our opinion, relatively insufficiently analyzed, and our data demonstrates the presence of novel infectious agents in most of these products. A species-specific analysis are scarce. I know of two of them who came up also with an increased risk for beef in that case, and occasional previous reports on involvement of other infection factors. There were reports of papillomavirus, polyomavirus, and a couple of others in colon cancer. They have not been reproducible. In our lab, not reproducible, but also in many others, and so they are not seriously considered. The protective effect of prolonged breastfeeding and of multiple deliveries have been mainly interpreted in the literature as hormonally determined. Our interpretation is indeed that specific sugars block the receptors for BMMF, here in this case. And our data provide also an explanation for the low risk of chicken, which was also investigated here quite intensively by the Vaki group, demonstrating that chicken have the same deletion as they do not produce new 5GC, so they do not provide the receptors for these types of infection. And also in fish, we have rearrangements of fish are frequently negative for these types of receptors. And the preventive effect of the non-steroidal anti-inflammatory drugs results from blocking the radical formation under those circumstances. So what we state here is that we identified a novel class of human pathogens. We call it tentatively the pathogenic plasmidomes. It's really a bovine plasmidome, which we only investigated so far. Linked them to specific cancers, colon and breast cancer, and we propose to name those cancers which are linked to those infections as plasmidosis, which needs to be discussed, of course. It is likely that several of these agents are able to induce similar persistent infection and chronic inflammations and the production of radicals of oxygen and nitrogen radicals under those conditions. And we suspect that members of this group also play a role in other cancers where these kinds of non-steroidal anti-inflammatory drugs play a protective role. And besides these cancers, which I mentioned before, in particular colon, breast, and prostate, other interesting candidates are chronic neurological diseases with inflammatory reactions, and that involves almost all of the chronic inflammatory diseases, besides multiple sclerosis, also Parkinson's disease, also Alzheimer's disease, and also amyotrophic lateral sclerosis as well. So that needs to be investigated. We have some studies going on along those lines, but the data are clearly not sufficiently advanced to be presented. And the other interesting aspect is that besides these diseases, are also other autoimmune diseases, and in addition also vascular inflammations in early stages of atherosclerosis deserve further analysis, because there you see the CD68 positive inflammatory 40 published in the internet at least In Google, you can find them if you look carefully into those data. And this should, of course, the analysis of those agents should permit the development of novel strategies for cancer prevention, diagnosis, and therapy. Prevention, in particular, by the development of vaccines, which most likely will have to be given to newborn babies during the breastfeeding period, like it is done for hepatitis B in many instances, particularly in Southeast Asia. For diagnosis, it's already available right now. And for therapy, we have the crystal structure of the Rep protein, at least analyzed, and theoretically, at least, this offers one possibility to look into potential chemicals which may specifically target this type of structure. So we understand at this moment why immunosuppression does not increase the rate of colon and breast cancer, quite different from the virus-linked cancers, the typical virus-linked cancers, because here we avoid, by immunosuppression, the inflammatory events in those cases. We prevent the development of oxygen radicals and mutations under those circumstances. Indeed, in the early phase of AIDS epidemic, the AIDS-infected women had a reduced risk of breast cancer in comparison to the non-infected ones. Later on with the therapy, it leveled off by now, but still not an increase. The members of this, as I said before, this infectious bovine plasmidome represents a novel class of human pathogens causing, as we propose it, plasmidosis. They emerge as main factors for cancer of the colon and the breast, and possibly for quite a number of others. The red meat of colon and breast cancers, commonly considered to be due to chemical carcinogens arising after roasting, broiling, barbecuing, and processing red meat, requires a revision. That is, for us at least, sure that it is required. The role, if there is any, is very minor and questionable. We understand the protective role of predominant chicken consumption and also of fish under those conditions for these types of cancers. And sugars in the milk, here I call them infectious bovine plasmidome receptors, play really an important role. The non-steroidal anti-inflammatory drugs prevent the events which I discussed before. So these infections emerge as promising candidates for cancer prevention, hopefully also for therapeutic approaches to treat these very common human cancers. The question which remains open, whether other ungulates, yaks, bisons, zebus, water buffaloes, sheep and goats, for instance, carry the same or related plasmids, derived structures, that remains to be studied. In Germany, at least, it's almost impossible to study it because our veterinarians do not permit the import of some kind of these sera, fearing the outbreak of foot and mouth disease due to our laboratory analysis, which I think is a little bit far-fetched, but I understand on the other hand also there are problems. There's a group which works on these whole problems, a very motivated young group of, at least in the center, and the other ones are a little bit older, which are division heads like Heikenfeld at ours and Brenner at our center, the German Cancer Research Center, and the two people from Stockholm, Olson and Grönland, helped us tremendously in getting the multiple sclerosis, well-characterized multiple sclerosis sera. Wolfram Scheuren, who helped us already in the early 80s in collecting some of the biopsies which we investigated later, and Achim Weber, who as a pathologist in Zurich was quite effective in providing us with beautifully characterized samples. So in a way, it's a long way. These are our involuntary collaborators, but they are very important for our work, essentially really for our work. So I'm gladly showing them as well. These are exactly the cause from which the blood was being taken later on. Okay, thank you for your attention. Thank you very much.